Thanks very much for having me. So again, I apologize that I'm going to have to leave a little bit early. So either if you don't catch me today, go ahead and email. So what I'm going to do is talk really fairly broadly about this topic, and I want to bring in a lot of sort of the challenges that we have both in data generation and data analysis to sort of help set some of the stage. We've had the really good luck to work closely with Nils and his team and Katja and her team, as well as Robert Kruger, who's here, and others, and very much think that the visualization challenge and the analytical challenge are the primary ones in this field. I should start by just mentioning that I'm on the boards of a couple, mainly the SAVs of a couple of the companies whose technologies I'm going to show. And we've been really lucky over the last few years as we move more into 3D, where the microscopes tend to start at about $1 million that they've been loaned to us because we can't necessarily afford them. So what I'm going to talk about today is really two aspects of this problem. One of them really has to do with sort of discovery, and that's where we see 3D initially playing the role. It's self-evident, I think, to all of us that medical imaging, which typically means radiological imaging, PET, CT, et cetera, those are inherently 3D methods, tomographic methods. But in clinical practice, essentially all data is collected from 5-micron thick tissue sections. They're occasionally frozen, particularly intraoperatively, but primarily they're formaldehyde fixed, paraffin embedded, as most of you will have encountered, so-called FFPE specimens. They're cut 5 microns thick, and then they're imaged. And as I'll mention, this is the primary diagnostic modality for many medical diseases and absolutely for cancer. So 95% of all cancers are diagnosed histologically. So I think one of the motivations of this type of tissue imaging is to try to impact the way histopathology is practiced in the clinic, and I'll allude to what that might be along the way. Now, at the moment, we have these two fields, sort of the spatial omics field that we might be representative of, and this more traditional field. There's also, as I'll mention, quite a lot of interest in the use of machine learning algorithms directly at the pixel level on these H&E fields. But our two parts of this, the more clinically focused one and the research one represented here today, are actually surprisingly disconnected. So, for example, I was a keynote speaker at the Association of Molecular Pathologists recently in Vancouver, the second largest molecular pathology group in North America, and there was no spatial biology. So it's sort of an interesting sort of schism there, and I think it's one of the things we'd like to address. And the reason for that, in part, is that we'd really like to impact those kind of things like diagnosis and precision medicine, et cetera. And so I'll touch on how some of that might practically be accomplished. So there's a fairly large spatial biology toolkit that most of you are aware of. It certainly includes the high-plex imaging that I'm going to talk about primarily today, spatial transcriptomics, which we integrate. A primary difference is about 20-fold in cost. So spatial transcriptomics currently is a fairly expensive technology. There's the H&E imaging to which I alluded. There's mass spectrometry imaging that I'm not sure we're going to talk about much today, but is extremely interesting because at near cellular resolution, it can look at everything below 3,000 to 5,000 molecular weight, which means metabolites and drugs. So it's going to be a very interesting way to look at drug distribution in treated mice or patient specimens. And then draw to your attention work from Brian Brown in Mount Sinai on a method called ProCodes, which is a way of actually doing in situ CRISPR screening in animals. It's a mouse method where you can put up to 40 or 50 different CRISPR clones into a single animal and then encode that spatially. So you can actually look then at the spatial distribution of CRISPR knockout, and interestingly, how clones would interact with each other. And I won't talk about those in detail today, but just to bring them to your attention. So the kind of problem that we've been tackling in the lab over the last few years really has to do with tumor progression and tumor initiation. So we increasingly find ourselves dealing with this question of how do we intercept cancer a lot earlier, because it's at the earliest stages that it's typically curable. So to give you an example here for melanoma, this progresses from an initial lesion, and by the time you get into your mid-60s and you have fairly light skin, you're going to be carrying hundreds of oncogenic mutations per square centimeter of skin, but those don't progress into cancer because of active immune surveillance. So the tumor not only needs to develop a program of invasion and excess proliferation, what we think of as the cell intrinsic factors, but it has to suppress the local natural immune editing. And this will then lead to a stage progression, eventually leading, in the case of melanoma, as many of you know, to metastatic spread when it's fairly small. And what's interesting about this side of the problem is that if you can catch the tumor when it's still only a few millimeters, you potentially have a curative resection. So a small, relatively low impact surgery can be curative. But among that small number of possibly curative therapies, we now know lie the born bad tumors. And the born bad tumors are the ones that go on, even with early detection, to lead to distant metastatic spread, which even today with introduction of immunotherapy and small molecule therapy is typically a lethal diagnosis. So a lot of what we're trying to do is really capture those tumor states early. And that actually contrasts with almost all the data that you're going to get out there in disease, particularly cancer, which is late stage. So 99% of what's out there in terms of genetics and genomics and anything in cell lines is actually looking at late metastatic disease because it's more obvious, it's easier to acquire. But for many of us, the big opportunity lays in this early detection, early diagnosis space. So one of the things that comes to the fore as you begin to work on this is that specimens as acquired from the clinic typically contain multiple disease stages. And so this has led us to realize over the years that an absolutely essential first step, and I'm going to come back to this several times in my talk, is histopathological annotation. And so what you see on the top here is an H&E, the pink and purple image, been around since the late 1890s, invented like must of histopathology by a bunch of creative German chemists. And that is still the dominant modality. So this field has not actually been, and to this day at Harvard hospitals, most of this data is not digital. Everywhere else in the world it is digital, but it's been fairly slow to become digitized. You could imagine, and in fact this is the case, pixel level learning on these types of images could be very valuable, and you've seen a lot of these foundation models come out over the last few years. What's interesting is how poor the labeling is in these foundation models. So if I show you the specimen here, this is a melanoma in situ with a precursor, so that multiple stages that I showed you before, and the vertical growth phase melanoma, about 1 millimeter thick, is what the diagnostic label carries, and that represents fewer than 2% of the total cells in this specimen. So the diagnostic label doesn't really capture the diversity. And so this represents this thing we think of as top-down analysis. And so this group is probably committed to doing these things quantitatively and from bottom up, but I cannot recommend to you more the value of 150 years, and we estimate 2 million publications in histopathology that have worked out what are the critical recurrent features in disease. And figuring out how to now capture that knowledge in a computable form, I think, is one of our challenges. The other way, which is perhaps familiar to you, is to try to build spatial neighborhoods up de novo. Here's an example of a fairly widely used method. This would be latent Dirichlet allocation, so drawn from language processing, and just looks for recurrent neighborhoods based on the statistical features in proximity. Remarkably, top-down labeling as done by a histopathologist and bottom-up labeling, which we both do, meet each other extremely rarely. And one of the reasons, of course, is that if you think the default for a tissue like our epithelium is a long sort of stretch of cells attached by their edges, that is a very low entropy state, right? That is a very unlikely state. And most of these models still have embedded within them the concept of radial symmetry, and a break from radial symmetry is the statistically significant event. So really interesting challenge, I think, with respect to analysis, and I'll come back to it. So let me just give you an example of this. So this is actually a couple of channels from a high-plex tissue image. You can see this is a couple of hundreds to 1,000 cells shown here based on the spatial scale. What you're looking at is a thumb of invasive melanoma, which is in blue, sticking down into the dermis. And in this case, what we were trying to find was a significant environment that was associated with tumor immune interaction. And let me show you what that environment turns out to be. We found it by human annotation. We went through and drew the edge of the invasive tumor. And let me point out, in passing, in oncology, actually finding the tumor is remarkably difficult. Most machine learning algorithms still can't do it, and there is rarely a definitive molecular marker. So we're often dependent on human expertise in this case. So we're now integrate along this barrier, and what we found is there's a local environment that's 3 cells wide. And you can sort of see that represented here with just a couple of the channels. And I'll give you one more set. So this shows that we have a rapid fall off in the tumor compartment, a rise in the stromal or local environment, and that there will be expression in this case of critical immunosuppressant genes that is 1 cell thick along that edge. And if you now take that piece of knowledge and you integrate it into a gene expression profile, and we've done this by a micro-regional approach, you can actually see statistically significant upregulation of gene expression proteins in this particular environment. So there's a whole bunch of biology in here that I won't take you to. You know, it's biology when it's a bunch of circles with arrows. But what's critical, I think, for this audience is to realize how tricky such a kind of significant microenvironment is to find with any of our current computational tools, just because of its relative improbability in the absence of prior knowledge. The other thing that I want to point out about these data, and this is particularly in the case of oncology, but is actually arising in other inflammatory diseases, and we're seeing it in infectious disease as well. So here what we've done is we've taken about 50 to 80 of those early melanomas, and we're sort of analyzing them at single cell and micro-regional level. And what you're seeing on the left is just a gene expression profile program using the same technology before, so-called geomics, which pools a couple of hundreds to a couple of thousand cells. And what we can see and then we put it onto a simple PCA plot here. We tend to try to start with regression and sort of end with ML. I know that that's backwards in modern practice. But here what you can see is that there is an overall trajectory shown in green, sort of indicated by the little dots there, where we go from the least developed tumor, so the so-called precursor field, towards the more developed primary tumor. But what's striking, if we now go to a single specimen now mapped out onto that PCA plot, is single tumor specimens, and we're seeing this also, as I said, in other diseases, can contain as much variation within them as we see across the totality of a patient cohort here with different clinical outcomes. And so that really is this problem here in the oncology case known as intratumoral heterogeneity. And it represents, I think, a major challenge, but also a major opportunity as we get to 3D, because the question is, how much of the specimen do we need to sample? And for those of you who have seen sort of the practice, particularly with first generation imaging technologies of using what are called tissue microarrays or little dots of things, I can tell you that that is statistically insignificant for almost any feature that you would be interested in, because with respect to spatial scale, one of those little dots is actually smaller than the feature you're trying to find. And of course, that means that you're dealing with statistical significance under spatial correlation, which is completely different than sampling random cells from a tube. And so again, a very unfamiliar kind of problem to most of us, and one that really demands a lot more thinking and analysis. And the consequence of this is that most of the papers that are out there today that are looking for disease-associated signatures are in fact statistically underpowered to find. And we'll come back to that in a moment. And here's just a little example of this kind of intratumoral heterogeneity. This is a single patient specimen. It has a couple of different disease stages. And what I want you to see is two things here. One on the left, that you can actually see dramatic, and these are, by the way, sort of differentiation-associated proteins. Some of them are used diagnostically. And you can see that over this specimen, which is a couple of millimeters, you're having sort of a dramatic difference between green and red. Both of those are tumor. They would look roughly the same in H&E. But also when you drill down at the single cell level, you can see that individual cells differ one from the next. And we might think, we haven't in general proven this, that the left side might represent a genetic change, but the right-hand side almost certainly represents epigenetic changes, or what is called tumor plasticity. So this ability of cells to adapt to and reflect the properties of their microenvironment. So let me just give you one last example of that. So this is just a couple of markers. Don't worry about the details. But what we're looking at is two things here. We're looking both at the trend across a series, and that's the blue lines. But the thing we've become more and more interested in is the variance within the data set, and that's thrown by the red lines. And so in some cases, as you go forward with disease, you don't see a significant change in the median value of one of these extracted features. You see a significant change in the variance of the features. And for my lab, I think for the last 20 years, has been sort of pushing on this point. It's really this issue of variance within the data that is one of the most striking features of the dysregulation of human biology, rather than this A versus B comparison. And I think many people here with the visualization haven't gotten to that, but you'll see every single paper. I challenge you to look at it. You'll see A versus B comparison. They get to statistical significance, which in medicine is always 0.05, no multi-hypothesis test unless it's genomics. But you'll actually look and you'll see the variance within the data is 10 times bigger than the effect size between data sets. And so that's something that we consistently see in these types of data. So here's just an example of that, where we look at two different modalities. And now begin to think of the visualization problem here. Do you look at one piece or do you look at another piece? In a weak moment, I suggested to one of our program officers that all we had to do was actually collect one disease specimen at scale, and then whatever hypothesis you had, we could zero in onto that region of the sample that showed the hypothesis that you needed to get your figure 3 paper in Cell. And that is why actually releasing whole slide or whole sample data sets, exactly what this community is committed to, and getting away from what microscopy has been for the last century, which is the postage stamp. So we have in our lab $1 million microscopes, and we publish 200 pixel by 200 pixel postage stamps of the typical image. And the typical image means we saw it once, and it met the hypothesis of reviewer 3. So consider this data set. You wanted to show that pink was important, or green was important, or yellow was important. And the answer is you could make the data look that way. And the problem is not that people are misleading themselves in these cases. Sorry, excuse me, misleading us. They're misleading themselves, right, by the way the data is analyzed. Now, one of the things that we've encountered in this is that what I've just talked about intertumoral heterogeneity doesn't seem to fit what we know from gene expression studies, in this case of tumors, that have come out from, say, single cell sequencing. So the Xenium data set here is a single cell picture out of roughly 450 genes. It's probably the leading single cell transcript profiling technology at the moment. So it allows us to address this question. What you see on the left is a high-plex protein image with a couple of colors shown, and this is a metastatic melanoma. So if you actually look in, as I mentioned before, we have that sort of speckling at the single cell level, and that's this heterogeneity we've been talking about. But if we now go into the Xenium data, the left is a clustering done on the gene expression. I'm sort of skipping along here, and there's a UMAP on the gene expression profile. And we can say that those differences between yellow, green, and blue and red there are sort of significant with respect to gene expression profiling, GSEA profiles that are out there. What is remarkable, I think, is yes, this heterogeneous tumor that I'm talking about is in fact organized in terms of gene expression space if we took all the cells and ground them up and threw them in a tube. But cells that come from different parts of gene expression state are in fact neighbors with each other, exactly as the protein data would show. So therefore, that's sort of the reconciliation between the dissociated bulk view, namely that there are discrete gene expression proteins, and this spatial view, where remarkably they live in immediate proximity to each other. And the easiest proximity to think about is their different cell types, right? One is an immune cell and one's a tumor cell. What I'm showing you here are all tumor cells. So tumor cells live in these completely different states for reasons I think we do not fully understand in immediate proximity. And working out how to move back and forth between these two types of visualization, which is very much what Nils showed before, between sort of the data visualization and true 3D, and then the derived feature representation is a really big task. And Robert, for example, and others has built some tools to do that, but doing that in a facile way is still pretty challenging. So I want to show you one other example of this, and this is actually our work on trying to get at, so people have said to us, well, you know, that's interesting, but if we actually go to something we really care about, like a therapeutic response in any single patient, it's probably much more likely to have a kind of uniformity to it. So I want to show you an example of this that we've been doing in the case of immunotherapy. And so in the last couple of years, there's been a fundamental shift in how immunotherapy is used from what's called, and many of you may have heard this, from what's called the adjuvant setting to the neoadjuvant setting. So adjuvant setting is in the case of a tumor that's resectable, surgery first, drug second. Neoadjuvant is drug first, surgery second. And it's turned out for immune checkpoint inhibitors, immunotherapy, that neoadjuvant actually gives better responses, which is actually contrary to many years of thinking, which was that you needed to debulk the tumor with a systemic therapy before you did surgery. But from our perspective, it leads to this really interesting feature that now we can routinely get tumors resected from patients that have undergone a couple of cycles of therapy. So we can actually look at, and they will usually have had a diagnostic biopsy earlier. So we can get this pre-post look. And I just want to show you an example of one of these. This is in stage 3 melanoma. So it's just spread local regionally to the lymph nodes. And these patients have been treated with several cycles of immune checkpoint inhibitor. And this is what a specimen like that looks. We're only looking here in 2D. We'll be getting to 3D in a moment. There's a necrotic core in the middle. So that's where all the dead cells are. So it tells you that this therapy was likely partially affected. And then I'm just going to show you 2 regions that we've chosen. This is Shashir Pant's work. And then we're going to zoom in on them. And the question is, what did this therapeutic response look like? And just with these little boxes in mind, we've saw a couple of mechanisms of killing in here. So what you hope to see is with immunotherapy is T cells have been activated. It's possible also B cells, and that those actually go on in a cytolytic capacity to actually kill the tumor. And we can see 2 examples of this that I won't take you through in detail, but where antigen presenting cells are partnered with cytotoxic T cells and B cells. However, in exactly the same tumor, depending on where we move our little sort of picture around, we can now find at least 4 different mechanisms of immunosuppression going on. And so the immunosuppression here can be of either of 2 types. Remember, our immune responses come into the tumor initially or into an infectious disease and then need to be downregulated. So you have both the natural downregulation of the tumor response and, of the immune response, and you also have the tumor attempting to suppress it. And that is, I'm showing you this to, again, point out not just the heterogeneity, but the fact that in almost all of this type of imaging, we have only a single snapshot in time. And one of the big challenges now is to try to figure out how to use things like temporal reconstruction or pseudo-time reconstruction, et cetera, to begin to get at the temporal series of an event that's going on here. And that's where I think increasingly mouse studies are going to be valuable to us, relating them back and forth to the humans, because in a mouse, you can get a more nearly synchronized response. But we don't have such data. The most valuable of all will be to go to intravital imaging. I don't know how many people here have encountered intravital imaging data in a mouse where you actually can watch cells in real time move around in a tissue. Those are typically 2 to 3 color images now. But if we could now fix those specimens and then subject to them to the methods that we're using more broadly, we could actually draw that trajectory in time, at least inferentially. So I think the big issue here is really this, the issue of the famous elephant here is depending on how you want the mechanism to work, you just look at a different piece of the mechanism and a different piece of the elephant. And part of the issue, as I've alluded to, is to try to work out what that heterogeneity means for an actual patient response or for a biological mechanism, and also what it means in terms of a temporal trajectory. I was going to say a word about mice, but I'm not going to do that. There's a lot of interest in mice. What we tend to think of now as mouse models is each representing one piece of it. So in the last couple of years, Clarence Yap, who's here today, and his colleagues in the lab, Alex, is shown here, have begun to move away from all the 5-micron data we've seen today. And I'm going to come back at the end and tell you why we should not disparage it, because it's going to be available in volume, and 3D imaging is going to be available at a much smaller volume. And in fact, the most recent 3D imaging data we now collect has to be sneaker netted around. So we can no longer, these data sets are so large, we can't transfer them on the network. So there are all sorts of behind the scenes challenges with that. And you should ask Clarence if you're interested. So one of the things Clarence and Alex and others have done, Daniel Yee's here today is also working on, a graduate student in the lab, is to go through and now take much better, much more advanced microscopes than are typically used in spatial profiling. So it's an irony that this problem of doing cell biology on human tissues in the dish is one of the hardest problems we've actually encountered as microscopists, and we use probably the worst microscopes available to do it, these sort of tissue scanning, low resolution microscopes. So here what we're using is a high performance confocal microscope. I'll show you a bit about how it works. But one of the first things we discovered was that in the 5 micron sections that most data, including most 3D data, is made up from now with serial section, no nuclei are intact and neither are any cells. And so you can see that by the sort of slices of the orange. If you go in and now use high resolution imaging, these are now just some reconstructed pictures, and this is a mixture of pixel level data and the meshes that Nils introduced before. You can actually see a level of detail in individual human cells, that's what we're looking at here, that have been 5 years in somebody's closet that you would not otherwise have noticed except in tissue culture experiments. So the capacity to push resolution here is largely unrealized. And resolution is valuable in this setting because it allows us to look at function, it allows us to look at morphology, and remember, spatially morphology is a key feature. It's not just genes on an XYZ grid. And it also allows us to improve signal to noise. So all of those things get better at resolution as this is sort of illustrating. So let me just give you an example of one of these. You can also begin to do some 3-dimensional reconstruction. So this is an early stage melanoma. This is data that I think Nils's lab has had access to. We've been trying for a long time to publish. And this is a 3-dimensional reconstruction of a section of a human melanoma in 56 channels at 200 nanometers resolution. And what we're looking at here is now a rendering of a single venule. So a small section of the blood vessel here. We know it's functional because you can see there actually is an RBC in there that's floating through the through this little piece of arteriole, which has probably been cut off by the sectioning. And here's just an example of a B cell. And if you've ever seen a B cell in a textbook, they're always round. They're the classical round cells. But of course, they're not round in tissue because in this case, they're undergoing transendothelial migration. They're going from inside the blood vessel to the outside. And so in so doing, they have to dissolve their skeleton, cytoskeleton, and their nuclei. And you can see that there still is a myeloid cell in here, so a macrophage or dendritic cell that's actually moving along the vessel. Here's just sort of a representation of that, but I won't take you through it in detail. The other thing we're able to do now in 3 dimensions is actually begin to build networks of cells. And so here's sort of a network of cells. And one of the things we've realized in this kind of 3-dimensional data set is that we're missing most of the cellular interactions that are visible in a tissue when we go to 2D, because we've chopped whole sections of a single cell apart. It's not just that we didn't see multicellular layers. We're not even seeing what's happening in a single cell. And here's an example where we've mapped for one little community of cells. I'll show you another one in a moment. And what you can do in these microscopes is begin to go down to resolutions. You can infer resolutions down to the size of a single signaling complex. And that would be around 30 to 50 nanometers. And that allows you to actually now begin to functionalize things like receptor ligand complexes. So from a visualization standpoint, the challenge here is we're looking at problems where there might be 10 to the 9th cells, so a centimeter or 2, and yet we're trying to go down to features that are subcellular, so around 4 logs in terms of dimensionality. And you can imagine that data set is going to have to be dynamically loaded and visualized. For us, this was one of the most sort of interesting experiments that we did. So what you're looking at here, we've isolated from that 3-dimensional tissue, we've isolated 3 cells, 1 dendritic cell and 2 T cells, and we just blanked everything else out for simplicity. We're not looking from the top, X, Y, we're looking actually from the side, so Y, Z. So the vertical axis down the slide here is the axis of the microscope. So you can see that's what a 3D image looks like. And it's accomplished in these type of microscopes by optical sectioning. And the yellow, sorry, the blue on the left shows you how many optical sections we took. In this case, around 130. So we move the specimen just a tiny little bit. It's all done automatically, of course, little tiny bit, and then we collect another image one after another, and then you do this 3-dimensional reconstruction. Now, what we can do, and what a conventional microscope do, would just take a single slice, and its image plane is shown there in blue. So, by the way, as you go 3D, 3D means high resolution means 3D. So they're actually conjugates of each other, except under special circumstances. The other thing you can do is then make virtual slices out this. You can say, what would I have seen now that I have 3D if I did 2D? And so we'll go and make a bunch of virtual slices, and then we can blur that out so we can actually now recreate virtual 2D data of the type most of us use. And this particular mapping here has been done with a physical method, which is not very good. And we're right now in the middle of building GANs that allow you to actually directly transform from different types of modalities. And I'll come back to this later. The reason for that is we think that even if it's available only in somewhat limited form, high resolution three-dimensional data is going to be the gold standard we're going to use for cell segmentation, tissue type identification, et cetera. And what we have currently in 5-micron data is sort of a tin standard. So, we tend to have in these problems bronze standards, and you say, here's the human annotated standard, how good is my machine learning algorithm? But if the human standard is already really problematic, then you have a real difficulty getting algorithmic convergence. So here's a case where we're going to go through, we're going to just take that thing, we're going to slice it into a bunch of different slices. We can then blur them or not, and we can ask, what would we have seen of this particular set of 3 cells if we'd only done 2-dimensional imaging? And in one case, we'll see that, and here are just sort of 3 exemplars. One is we actually misphenotype the cells altogether. Because remember, a good fraction of the proteins on cells, and possibly also genes, are not expressed uniformly. As part of their function, they're highly polarized, for example, to the top edge of a cell. And if we cut that off, we just don't know that they're not there. And phenotyping, as it's done in all of this space, is like flow. It's a plus-minus signal. So we use the absence of information as a measure of its cell type. The other thing is, in many cases, you're going to actually get the phenotype wrong because things in 2D, you're getting a projection of different cell types lying on top of each other. I guess it'd be this way around. And we project them down. We say, well, that signal lives inside of this cell, but we're wrong. And actually, that is why cell segmentation is so inaccurate in part. It's not an algorithmic limitation. It's a fundamental data limitation. And the famous case, if any of you have encountered it, is the cytokeratin-containing T cell. No T cell has ever contained cytokeratin. And in the segmentation data in H10 elsewhere, about 20% of T cells have cytokeratin, and that's because here's the T cell and here's the tumor cell, and we've projected them on top of each other. And that is not going to be fixed in the current standard just by small improvements in segmentation. And then there are other things of interest more technically to us. We always have background signal in these things, and everyone frets about it. And now we realize more and more background signal is actually real signal from the cell above. But if we didn't capture the nucleus, we didn't know that it was there. So both technical, I think, and scientific insight. Let me just show you one of these multicellular environments here. And what we're looking at is one in which, I'll just go back for a second, one in which we actually have multiple things going on at the same time. So the way we tend to think about immunology as it's been reconstructed in vitro is you'll have this temporal series to which I alluded. You'll come in, you'll be activated, you'll kill a cell, and you'll be repressed. So I want to show you one T cell here that's doing all of those things at the same time. And it gives you a little bit of a sense of the data. So again, this is one of these 30 to 40 micron data sets. It's in 50 channels or so. We just peel those channels back to make it a little bit easier to see. You can see we're applying a rendering engine here. We're independently calling tumor type and tissue type, and some of this is done automatically, and a good fraction of it is still done using the ibolatron, which is that you get a couple of beers and you stare at this late at night. So big opportunities for improving segmentation. And here's an example where we see a T cell that carries one of these so-called activation exhaustion markers, LAG3. Interestingly, it's all intracellular, which has important applications. We can see that in this case, the killing enzyme, which is granzyme B, is actually polarized towards the tumor cell. So here's a T cell directly killing a tumor cell as best we can understand it. And the most famous of all the immunosuppressive events on a T cell is PD-1, PD-L1, the target of immune checkpoint therapy. This exact same T cell is undergoing a repressive interaction, and it's also undergoing a helper interaction with a third neighbor. So all 3 of the prominent features of the progression of immunology can be seen simultaneously in human tissue. And so I think this is part of the promise for our field to rewrite the way we think about tissue biology. And remember, tissue immunology. Remember that about a third to half of our immune system lives in our tissues, and the skin and the gut are prominent examples. I mentioned this before, but if we think about the segmentation challenge here, the way it's conventionally done is to use a microscope as though it was a really low quality flow cytometer. And so that's one of the things we absolutely have to get away from. So what we do is we integrate cells with some sort of a smoothed mask. Cells are not smooth. We then do intensity-based gating. We either hide it in the methods or we're upfront about it. We may do high-dimensional clustering, which is maybe a little bit better. And then we enumerate cell types. But as I've just shown you, these cells are completely intermingled with each other. So this is really not going to work. And so I'll send that one to you. And so what we've begun to work on is new approaches. And Greg Baker, who's not here at the moment, is an example, which is actually to just do what a human does. So it's interesting, if you give a human a tissue and you ask a couple of either trained pathologists or also tissue biologists to begin to call T cells, their inter-observer agreement is much better than the performance of current algorithms. And that's because they're looking at three things we now understand. One is intensity, the second is morphology, and the third is neighborhood. Because implicitly, when you look at a tissue, you're seeing its neighborhood features. And we've now begun to build machine learning tools that can do that. And one I can commend to you that's just coming out from Greg Baker uses VAEs, variational autoencoders, an old sort of fashioned type of machine learning method, but one of whose beauty is you can look in the latent space and pop out exactly what's there. So here's sort of a kind of fanciful image that Greg has put, where he's done a UMAP, where he's overlaid on it the VAE embedding for each of the cells that's there. And you can begin to see that this type of thing is not capturing that it's orange alone, it's capturing that it's orange, it's approximately round, it's an immune cell in this case, and it's got a hole in the middle because it's a cytoplasmic signal. So primitive though this is, these kind of tools look like they're going to dramatically improve the way we do segmentation. But there's really very little development in this area. So another area where algorithmic development, not visualization per se, but is going to help us with building accurate meshes. The other thing we've begun to do is, and this is work of a graduate student in the lab, Gui Hong, is to begin to actually go back to those foundational models that everybody is talking about and actually ask if they can do something useful, like find tumor versus non-tumor. And the answer for the most part now is no. They actually can't do something that a human pathologist can do very well. But what they can do is find certain types of features extremely well. And we don't currently use the H&E data that is available to us because we sort of are like, purple, pink data, not so interesting. So I think one of the things we like to do increasingly is link in those models where single cell data doesn't help us. And so here I'm showing you an example of vessels with red blood cells. And here spatial transcriptomics isn't going to help us because there's no nucleus in these cells. And also because there's no nucleus, no modern segmentation mask will ever find them. And in fact, if you go back and take a high dimensional image of the type I've shown you, and you say, what fraction of the cells there are never identified by any contemporary algorithm? It's between 10 and 30%. And so actually this older fashioned type of H&E data contains a huge amount of information about cell types. Admittedly, it's sort of this type, but if we can recognize that with classifiers, we'll be able to add it to our single cell data set. And then the final thing we've now begun to do, and this was developed by Faisal Mahmood and others here, is actually to create routine three-dimensional virtual H&E data. So the way we do that is we actually take all the channels we throw away in a microscope, the autofluorescence, we stain with nuclei, and then we either use a GAN or a color mapping system to regenerate what looks like H&E. That H&E is not stained with hematoxylin and eosin. And the reason for that is, by the way, when you get to H&E, you're end stage. You can't do any more molecular analysis. And this now can be put into 3D. And so I think the first 3D tissue data set of which you're going to see routinely is going to be this virtual H&E. And we're hoping to release a substantial amount of that data in the next year, because it's actually pretty simple to collect. The hard part is figuring out what it is. So if we can now take that three-dimensional data and we can now use either the foundation models plus classifiers and automatically label this. And we can then overlay some of the high dimensional data I've just shown you. We have the possibility, I think, for a really interesting near-term win. And one of the things we've seen there, no surprise, is in 3D, things look completely different than they do in 2D. And we've just misappreciated often their shape. So sort of conclusions for this, and I'm going to wrap up with a final 3D section, is really no cells are intact in a conventional 5-micron. Thick 3D gives us some access to that, and that you can use the resulting morphological information to inform on function. Now, what about using any of this in diagnosis? I think we read that this is all going to become 3-dimensional. It's going to be hyperplexed. The answer is there is no currently approved immunofluorescence-based test beyond single channel for any diagnostic anywhere in the world. So the gap between current practice and what we're talking about today is pretty large. And the areas where people are interested often get broken into these two categories. One, what's called prognostic biomarkers, which means predicting disease trajectory. Is somebody going to get, recurrence or not in the cancer case? And what's called predictive biomarkers. And there's no absolute logical distinction here, except that this is how it's described. And predictive biomarkers are whether you're going to respond to a drug or not. And so the most important predictive biomarker that uses imaging out there is for trastuzumab, is for Herceptin. And it's been around for 20 years. The first targeted therapy is if a breast cancer will routinely be stained for the cell surface protein HER2 and then scored by a pathologist from a scale of 1 to 4, and that will determine if a patient is then going to be prescribed Herceptin. And that is, as many of you probably know, the absolute first breakthrough therapy of the molecular age. The second most widely used is PD-L1, so that negative regulator that we saw for T cells. And that is currently regarded as the least effective diagnostic ever developed. The inter-observer agreement is poor, and it has extremely poor prognostic potential. But in fact, it's been required by the FDA for a subset of cancers from the time of the introduction of immunotherapy. So we see the whole sort of gamut. Companies hate to introduce new diagnostic tools in general. It's considered to be the least attractive place to market a new product. And so everyone actually, when we talk about precision medicine, it has two pieces: measure, which is diagnose, and treat. And about 95% of all funding is in retreat, and a little tiny bit by mistake leaks over into predict and diagnose. And so what we have to realize is our community is part of that other thing, is trying to figure out who should get the drug when, which is really an immensely underfunded activity, and got a boost because if you call it AI, now the tech bros like it and they fund you, but then they get disappointed because they realize that's actually really hard to get any reimbursement for it. So then you see what we're seeing now. The AI companies come up, they build something plausibly good and they can't bill for it. And the one exception to that, if anyone's interested in that problem, is in radiology, where there are 350 approved AI-based radiology tools. But that's because radiologists have really good billing codes, and pathologists do not. So here's that sort of tumor staging in oncology. When you think about it, where's all the emphasis been in the last, and I'll take just 5 more minutes. Where's all the emphasis been in the last 20 years? Gene expression, gene profiling, unbelievably important. If you have a mutant BRAF cancer, you're going to go on a trajectory that's totally different than if you don't. But it probably counts for between 8 and 12% of all diagnoses. And as I mentioned at the outset, 95% of all diagnoses are actually done histopathologically using stains developed by Germans in 1850 with microscopes developed in 1890, and then 150 years of human expertise. And it's that CNN, that human CNN, that we need to now figure out how to capture into our modeling methods. So there are these two divergent views of where we're going to go. One of them is actually it's going to be super complicated. As I alluded to, it's going to be hyperplex, Visium, $18,000 per specimen against a standard reimbursement, by the way, of $12 per patient. Where we are is on the left. Or it's going to be all AI and no molecular features. And so that has been a big push, and that those circles, which you can't read, are all the companies that the tech bros have put together to give this a try. We actually think there's going to be a different intermediate, and by sort of being the intermediate, of course, you suddenly find neither side likes you. We think the intermediate is going to be to develop a set of systems. And here's one we developed through an SBIR, in which we actually do what we call optimized midplex multimodal imaging, OMI. And so the idea is we're not going to collect 100,000 channels diagnostically. We just can't. And we don't know how to process the data. We don't know how to normalize it yet. But we must collect the H&E as well. That's the multimodal piece. So we built a microscope that can, RareSight did with some of our help, build a microscope that can do 20-plex imaging at pretty good resolution, not the resolutions you've seen, and it can integrate that with H&E on the same slide. And so that's the type of data, either from this or other platforms, that we're likely to see coming in volume. And this leads then to the last sort of point about the computation is now we need to figure out how to divide, really bridge those worlds between what the pathologists of my knowledge call the sexyplexy, which is whenever it's more than 2-plex. So the sexyplexy and the H&E. And you can think of all sorts of tricks back and forth. Use the molecular data to label the H&E data, use the H&E data to segment the molecular data, et cetera. And there's been very little exploration. And I think for this community, the thing we'd be most excited about is getting that virtual H&E sort of embedded with the molecular data and figuring out how to calculate meshes on that. I think that actually could be quite an interesting near-term win. So we've done a little bit. We have a visualization environment that we've intentionally tried to make complementary to one that Nils has developed. We call it Minerva. It's meant to be ultra lightweight, so it runs with really no desktop software, et cetera. But one of the little tools that's been the most popular in it here is when you have a multimodal data set, you can drag these little windows around in a browser, and you can go from H&E, which a pathologist likes to interpret, back and forth between that and the molecular data. And this, to us, we found actually allows you to take 150 years of human experience and then what we call modern measurement technologies and bring them together. So I don't want to spend too much time on this, given the hour here. I did want to conclude with one point, which is we've actually done this now for human cohorts. And here's actually a cohort we have now of human colorectal cancer specimens. And so colorectal cancer is interesting in this setting because there's almost no cancer type whose origins we understand better than colorectal cancer. And none of those molecular features are used diagnostically. So if anyone here is over 50, you've been in for a colonoscopy and they take out your, it's fancy, schmancy, it's well reimbursed, so the office is really nice. And you go in and they take the piece out, and remember there's hundreds of genes we know a huge amount about colon cancer. That early colonoscopy, they will determine the period at which you come back based on how far it was from the entrance of the endoscope, so where you are ascending, descending colon, whether it was stuck on the wall or whether it was pedunculated, which like hangs off like your epiglottis, and how many centimeters it is, and that's it. It's a formula that you could put on the back of an envelope, this many centimeters, this far up, et cetera. And that's the totality. There's no molecular data in here at all. And in fact, actually, the genetic features shown here are really minimally predictive. And so just on a problem like this, we've now gone through and captured the immune sort of contexture of these tumors in a fairly reliable and reproducible way. And this shows you between now a Kaplan-Meier curve between blue and red on a whole slide image, the difference between people who will progress and those who will not progress based exclusively on the immune contexture. And so that's the likely first application of our field in a diagnostic setting is routine analysis of the immune contexture of tumors. And what we found to our surprise over the last few years is it's extremely poorly correlated with genotype. And so that represents an incredible opportunity, but it also means that most of the signal has once again been hidden in those A versus B comparisons because we've not been looking at intra data set variants. So anyway, I'm just going to skip ahead on this and given the hour. This just shows you actually one thing we have been doing is getting this data into cBioportal. So I don't know how many people know about cBioportal, but if you are at all interested in cancer genetics, it's the place to go. It doesn't have spatial data now. And so working with Nikki Schultz and Ethan Cerami here at DFCI and at MSKCC, you can now get spatial data directly out of cBioportal. And I think this is Robert's image that I shoved in there, showing that you can't get this in cBioportal, but you can get something simpler to that. So you can actually begin to get some spatial data. And would recommend that to this community as an alternative interface to something like the Cell Atlas interfaces, whatever, which are sort of based on the human body and morphology. This one's going to give people entrance to our data types through genetics. And if you go to a genetics meeting on this topic, it would occupy the entire campus. And here's how many people are here for the picture meeting. So we think that the way this is going to develop is we're still in the research phase. There's Clarence looking down an old-fashioned microscope. We're building the instrumentation out. Visualization is everything here, and it's extremely poor in general. We think this can go forward into clinical development in the next couple of years, and there are a couple of centers doing that now. But the big challenge is going to be to get the energy together to actually do a clinical trial for this. And a biomarker clinical trial costs around $10 million, which is, by the way, about a 10th to 20th of what an interventional trial costs, but the total amount of money is about 1/100. So the last thing I'll leave you with here is this last issue of the elephant. And this is our attempt now to create true large-scale three-dimensional data sets. So even the three-dimensional data set I showed you a moment ago was only 35 microns thick. And so the question is, can you get a couple of millimeters of human tissue visualized in a microscope and then actually analyzed at a single cell level? And the way you do that is with a light sheet microscope, which has been around for 20 years. And the company that we work with here has sold several hundred, and that not a single one of them is used, as far as we know, to collect data from humans. They're exclusively used to collect data from transgenic, or what I heard the other day, the administration calls transgender mice. And so I think this is one of those cases where we're taking an existing technology and moving it over into human practice. And it turns out to be very difficult chemistry and optics. And Alex and Clarence, I think, have really broken this open. So you're going to begin to see these type of data sets. And we're ready to hand this one over to you soon, Nils and Katja. And you will be happy to know it's around 200 terabytes. Is that right, Clarence? Yeah, so these are really big data sets. And what's interesting about them, and I'll just play it for you one more time here. What's interesting about them is that by going to the bigger spatial scale, we can see blood vessels, nerves, and all those things that are really invisible at small spatial scale. And so we think that the overall thing in the future could be micro CT, which actually can work at the centimeter scale, this which can work at the millimeter scale, and then the three-dimensional imaging that I showed you, the very high resolution that can work at the submicron scale, at the micron scale. And so those three embeddings, you're not going to have everything on everything. So we're going to need visualization that sort of jumps, sort of from like the continent to the city to the house. So let me just say that I think there's a lot of opportunity here to begin to take some of these high dimensional methods and begin to build ground truth. Sorry, Daniel, I skipped over your data set there that just shows how we're going to take the three-dimensional data, help build a ground truth for 2D. Happy to show you more of that later. And we think that trying to develop links between this sort of machine learning pixel level world out there that's been notionally diagnostic and our world that's very segmentation and sort of mesh and point cloud based is actually an enormous opportunity. So I would recommend that to you. Thank the people who did this. Some of what I've shown you has been published. Here are some of the fellows. I tried to recognize them along the way, particularly the work of Clarence and his team on three-dimensional imaging. None of this would happen if we didn't have pathology collaborators. And so our HTAN team at NIH, I think, had more pathologists than anyone else. And it's just astonishing what these people can see. I mean, they are the most trained CNN that you've ever met, stuff you just absolutely have never seen. And I think they're really ready to be engaged by our community. Fantastic group of individuals here, shown largely from Brigham and Women's Hospital and MGH, led by my colleague Sandro Santagata. And then finally, we have a pretty large data and software team, and this is only partly representative of that. And we collaborate a lot with Hans-Peter Pfister in the analysis, and I didn't show the visualization work with Nils and his. So thank you very much, and take your questions.